Good morning. Thanks to Prof. Ali Reza Mousavi Jahari's kind invitation for me to give this keynote speech at the 10th APOCPGA. It has been a great pleasure to work with him and the team as part of the editorial board of APJCP, Asian Pacific Journal of Cancer Prevention. The original title given to me was Cancer Chemoprevention and Pharmacodynamics of Gene Expression. Here, I would like to present a very specific area of the cancer prevention involving NRF2 antioxidant signaling, focusing on the reason why I think we are blessed by many dietary compounds that possess cancer preventive properties. So the subtitle is The Blessings of Antioxidants in Food as Dietary Cancer Preventive Compounds. Pharmacodynamics concerns the actions of the chemical on the organisms. In this context, we are going to see the benefits of these compounds on gene expressions in various settings. The work presented here was done in Prof. Tony Kong's lab in the Rutgers University. There is no conflict of interest. This is the outline of today's presentation. First, I will introduce some of the relevant terms in the field of cancer prevention, the history of cancer prevention, and some cancer preventive compounds that are commonly found in our diets. Second, in this talk, I will focus on oxidative stress and its relationship with NRF2 pathway. It is the major transcriptional regulator of the expressions of genes encoding phase 2 detoxifying and antioxidants enzymes, including heme oxygenase 1, HO1. The role of NRF2 in protecting a person from oxidative stress will be discussed using NRF2 knockout mice. And last, I will discuss some challenges in this area and the aspect that is beyond antioxidant response. What is chemoprevention? The term chemoprevention was first coined by Michael Spons in 1976 when he referred to the prevention of malignancy development by vitamin A and its synthetic analogs. Since then, chemoprevention has been adopted as one of the major tactics to modulate the process of carcinogenesis. Many years of study have proven that this strategy is indeed effective in reducing the incidence of cancer in the well-defined high-risk groups. Chemoprevention, by definition, is the use of pharmacologic or natural agents to inhibit the development of invasive cancers. The chemicals with a cancer preventive activity are referred to as chemopreventive agents. An effective chemopreventive agent should be able to interfere with the early stages of carcinogenesis and to eliminate pre-malignant cells before they become malignant. Generally, it is believed that it takes years for a cancer to form in human body. Since carcinogenesis is a multi-step process, it provides us a great opportunity to intervene before a malignancy is formed. As of today, cancer remains a terrible challenge for human health. Currently, the major mainstream of treatments are surgery, radiotherapy, and chemotherapy. Each of them has serious side effects. This is especially true for chemotherapy side effects such as nausea, vomiting, alopecia, neuropathy, etc. The global cancer burden has taught us a lesson that prevention is better than cure. How can we alter the carcinogenesis process? The next slide will illustrate this idea. This is a schematic illustration to show the multi-step carcinogenesis. Generally speaking, tumor development involves three distinct yet closely linked phases: initiation, promotion, and progressions. The initiation step is started by the transformation of normal cells into cancer-initiated cells. This happens when the cells is subject to carcinogen or radiation exposure, for example. These initiated cells undergo tumor promotion into pre-neoplastic cells, which progress to neoplastic cells. Chemopreventive agents can interfere with different steps of this process. Some agents inhibit metabolic activation of the procarcinogens to the ultimate electrophilic species or their subsequent interaction with DNA. These agents therefore block tumor initiation. They are classified as blocking agents. Alternatively, blocking agents can stimulate the detoxification of carcinogens, leading to their excretion from the body. Other agents can suppress the later steps, the promotion and progression steps of multi-stage carcinogenesis. They are suppressing agents. Some agents can act as both blocking and suppressing agents. So far, most of the chemopreventive agents are plant-based. Although phyto is Greek for plants, they are not only present in the plants. Some may be present in seafood, such as astaxanthin. A lot of them are being sold as functional foods, health supplements, or nutraceuticals. They are good for health. As we can see, it is a very big business in US, and NIH has concern to investigate their benefits or harmful effects. Most of them are actually antioxidants. I will share with you what they do after we review some of the compounds. They are rich sources of glucosinolates. When converted, they form active compounds, including indoles and isothiocyanate. Resveratrol, quercetin, camphorol, and petrostilbene are abundant in berries. The antioxidative properties of these constituents may contribute to cancer prevention. Quercetin and camphorol and petrostilbene all exhibited strong free radical scavenging activity in the DPPH assay. They also attenuated intracellular reactive oxygen species ROS levels when pre-treated individually and had synergistic effects when used in combination. In addition, the combination treatment significantly induced ARE, the antioxidant response element, and increased the mRNA and protein expressions of NRF2 regulated genes. Collectively, our study demonstrated that the berry constituents, quercetin, camphor, and petrostilbene, activated the NRF2-ARE signaling pathway and exhibited synergistic antioxidative stress activity at appropriate concentrations. This work has been published, and I will give you the reference at the end of the presentation. are the chemical structures of selected chemopreventive agents with ability to activate the NRF2 ARE signaling pathway. As you can see, they are diverse in structures. They look pretty different structurally, but they are all able to activate the NRF2 pathway. In other words, they are antioxidants. By the way, OT plus is a synthesized compound. As mentioned before, chemopreventive compounds can be found in natural or synthetic source. Before I move on to the pathway itself, I would like to mention something about curcumin. Over the past decades, curcumin has become so popular that one can purchase it over the counter. By now, many of us will be aware that there are many health benefits when one takes curcumin, a constituent of turmeric ginger family Curcuma longa. Here, I just want to highlight two pathways among many others that are well established. First, it is a potent anti-inflammatory agent. Acts via inhibiting nuclear factor kappa B and FKB, the key pathway regulating inflammation. Second, it is a natural antioxidant that can activate NRF2-ARE pathway, the key regulator for oxidative stress and phase 2 gene metabolizing and antioxidative stress enzymes. It is well recognized that curcumin activates NRF2-NRE pathway, leading to induction of phase 2 genes such as glutathione S transferase and heme oxygenase HO1. This leads to my introduction of the NRF2 pathway. It regulates antioxidative stress enzyme and phase 2 drug metabolizing and detoxifying enzymes by binding to antioxidant response elements. You will see it later. The cytoplasmic KEAP1 inhibits the translocation of NRF2 into the nucleus. The inhibition is disrupted when there are oxidative stress or chemical inducer to dissociate KEAP1 from NRF2 binding, allowing the translocations of the transcriptional factor NRF2 to enter into the nucleus and dimerizes with SMAF proteins, and eventually NRF2 binds to ARE to produce phase 2 gene detoxifying antioxidant enzymes such as GST, HO1, and NQO1, which are cytoprotective enzymes. They enhance the antioxidant capacity of an organism such as mice or humans, eventually prevent cancer formation. You may have a question why I am so certain that the NRF2 gene must be involved in cytoprotection against oxidative stress. This is because we have done experiments in the mice that are without NRF2 gene, which we call the NRF2 knockout mice, NRF2KO, and compare them with the wild-type mice, the mice with intact NRF2 function. All wild-type mice have NRF2. We as humans are wild-type 2 in this sense. So we also have NRF2 by birth. This is a schematic presentation showing that NRF2 disrupted mice have a higher susceptibility to carcinogenesis. ROS represents reactive oxygen species. RNS represents reactive nitrogen species, and both of them are stressful to cells. Thank God that we are all wild type in these regards. Otherwise, the cancer incidence would be much higher. Of course, it is unethical to do such experiments in humans, so we did such experiment in mice in order to have a better understanding on the mechanism itself. Taking the colorectal cancer as an example, there is a model we use to study mechanism involved, which is illustrated in the next slide. Here is a simplified illustration shows the carcinogenesis in human colorectal cancer, CRC, and the role of NRF2 in anti-inflammation and oxidative stress. Current studies have linked chronic inflammation to cancer development. CRC is one of the classic examples that demonstrate a close relationship between chronic inflammation and carcinogenesis. Treatment of animal with dextran sulfate sodium DSS has shown to induce colitis, a chronic inflammatory disease, whereas treatment of animals with both AOM, azoxystrobin, and DSS induces CRC. Schematically, in an animal colorectal colon cancer CRC model, carcinogen AOM can be used to induce the initiation of the cancer formation stage, followed by an inflammatory agent, DSS, to promote the inflammation of the colon, colitis, and eventually aberrant crypt foci ACF, and tumor will form. This is a figure to show the role of NRF2 in protecting inflammation-associated cancer, showing a potential strategy for prevention of inflammation-associated cancers. When the NRF2 signaling pathway is activated, activation of expressions of phase 2 antioxidants and detoxifying enzymes such as HO1, NQO1, UGT, and GST alike in NRF2 wild-type animals can reduce the oxidative stress, and it is postulated that NRF2 could also regulate pro-inflammatory genes and cytokines such as INOX, COX2, and IL-6 alike. And taken together, they protect the animal from developing inflammation-associated cancer such as CRC. In contrast, NRF2 knockout mice do not possess functional NRF2 and NRF2 signaling pathway after prolonged inflammatory and oxidative stress caused by AOMDSS. They have increased incidence, multiplicity, and size of all colorectal tumors, including adenoma and adenocarcinoma, as compared with wild-type mice. Furthermore, AOMDSS treated NRF2-KO mice had more severe colitis and higher incidence of prolapse, rectum, and bleeding than wild-type mice. Therefore, NRF2 pathway appears to mediate a strong anti-inflammatory response, besides induction of detoxification and antioxidant enzymes as described above. the beginning of our talk, we have gone through many compounds of different chemical structure that are available in fruits and vegetables. They are interesting to us because we know that they are natural antioxidants and supposed to protect us from oxidative stress because oxidative damage to DNA can lead to DNA damage and therefore increase the risk of cancer formation. Although we have learned from NRF2 knockout mice on the protective function of NRF2 and some of the downstream gene expression induced by some of the chemopreventive compounds, I have to admit that these pathways are not all that we know. The next slide will give you an overview of what we know about the chemopreventive properties. Interestingly, when you match them with the 7 hallmarks of cancer, they seem to provide some opportunity for us to modulate the process of carcinogenesis. This is the idea that I published years ago. I call it lock and key model for chemopreventive compounds, neutralizing the acquired capabilities of cancer, the 7 hallmarks of cancer, namely cell sufficiency in growth signals, insensitivity to anti-growth signals, evading apoptosis, sustained angiogenesis, limitless replicative potential, tissue invasion and metastasis, inflammatory microenvironment. Targeting at NRF2 is one of the promising pathways, as many of the chemopreventive compounds targeting NRF2 also possess other molecular targeting ability, such as blocking the initiation and suppressing the promotion and progressions of cancer, regulating hormonal signaling, inducing apoptosis in cancer cells, anti-angiogenesis, regulating cell cycle, anti-metastasis, and anti-inflammation, and most importantly, prevent the carcinogenesis as a whole. That's all for my presentation. I hope you find this talk interesting. If you cannot remember anything that I shared, just try to remember eating fruits and vegetables are good and they contain a lot natural antioxidant that can activate NRF2 pathway. I would like to acknowledge the support given by Dr. Tony Kong in Rutgers University and all the colleagues and collaborators when I worked there. Thank you for your attention. If you are keen for the details, please look out for the references. The major references are listed here. Thank you so much.